Hello, very good evening to one and all. Am I audible? Hello.
Yes, sir, you are audible.
Okay, so you can put in the chat box, right? So I am Dr. Narottam Sharma on behalf of Make in Turn and Learn to Upgrade Academy. I will be taking care of you for another 12 days, right? So you have selected this course from Make in Turn and Learn to Upgrade Academy. So we will be talking and discussing in details as well as some of the other aspects in cancer biology. So if you will be finding any sort of the issues or the glitches in terms of, just internet issues or this presentations or voice clarity, you can directly put in the chat box or you can open your mic. And for the smooth conducting of this program for another 12 days. So we will be as a team and definitely we will be interacting with one or two more faculties, my colleagues over here for some of the practical sessions and some of the live video demonstrations we will be providing to you, right? So in this sessions, we will be talking about the journal biosafety guidelines, as well as before going into the classes, basically, because this is the very first day. So I am Dr. Narottam Sharma, PhD in medical biotechnology, and I'm working as a molecular biologist, virologist, and oncologist. And we have, so many programs which I used to conduct as a resource person for Make in Turn and Learn to Upgrade Academy. So we are the laboratory partner of Make in Turn and Learn to Upgrade Academy, because Learn to Upgrade and Make in Turn, they are basically providing the world class training programs, especially in the fields of cell biology, molecular biology, virology, COVID, then genetic engineering, recombinant DNA technology, and forensics, and life sciences sectors, and pharmaceutical sciences, and all. So I just want the students to, means I know, I want to know whether you are from which background. So can you just put in the chat box, not your name, not your colleges, nothing, but your degree, as well as, from which field you are, like BSc biotechnology or MSc biotechnology in chemistry, so that I will get an idea like to what content I have to emphasize and what things I have to emphasize more for you, so that I can customize this program in such a way so that it will be, fruitful for you people, right? So, okay, BSc, okay, fantastic, fantastic. I'm getting so many responses over here. I don't want your college name and your name, basically. I just want your disciplines. Okay, so some of the students are from zoology, botany, pharmacy, life sciences, biosciences, microbiology. Fine. And okay, so we will be taking the sessions with interactions with you students only. And we will be talking now more about the things that okay, MS in cellular and molecular medicines. Great. So thank you very much, my dear students at the other end. So we will be talking and discussing on a single platform, which we will be utilizing for conducting of so many practicals, which is going to be coming up from our laboratories to you only. So there will be, the demonstration of the practicals also, as well as we will be talking about the cancer biology and genetic engineering in terms of oncology, what are the various advancements that has happened. So I think I am very much audible to all of you, and you can see me as well as my voice is clear, right? So that I can proceed further. Okay, so we will be discussing about when we are talking about laboratory works as well as other sort of things. The very much important thing that will come into the mind is basically the good laboratory practices and the other things that we are going to have we have to at least follow, and how we are going to do the GLPs as well as the other laboratory sort of things, and the practicals, how we have to perform in the laboratory. So we will be taking you on a laboratory tour also today, as well as before going into the lab tour, I will be showing you I will be demonstrating you basically the BSL2 labs, right? Biosafety level 2 labs with the BSL3 practices, how this virology and a molecular level of works can be done in the lab, right? So in this regards, we will be talking much more important exercise, that is biosafety. That is again very, very important, because today I will be talking about some sort of the cancer epidemiology, how it started, how many cases were there. Before going into into the deep knowledge of these subjects, I want to take you to the this good laboratory practices, as well as the good laboratory things, biomedical waste managements, GLPs, as well as what are the basics precautionary measures, and what are the guidelines we have to follow in the lab, right? So for example, you all are from the life sciences, applied sciences, from medical streams, from medical backgrounds streams. So definitely it is an important things for all of us to know, like how we have to work in the laboratories, right? So first of all, like what is basically the biosafety, and how why we require biosafety, and what are the agents of biosafety, and what is the requirements for the biosafety, right? So we will be talking all these things in details in this presentation. So any agents that can cause deleterious effects, for example, you are working on microbes, so microbes can be the deleterious agents, and which can produce the some of the effects in case of the humans, right? And toxins, some of the gases, some of the components that is secreted by the microbes, and some of the media, they can also be basically the agents of they can cause the deleterious effects in humans, right? So this is basically the good laboratory practices important wing, we can say. We have to at least know to what agents we should take precautions. So microorganism, toxins, and allergens that is derived from the organisms, and allergens derived from higher plants and animals, they can cause they can be I'm not, my dear students, I am not putting any of the video. This is presentation only. So I think all can see the presentation, and I am audible. Once again, I want to ask from all the candidates. So
Yes,
Shweta, I'm not showing any of the video, dear. This is just the PowerPoint presentation, right? Can you see? Okay, fine, fine. Okay, whenever I will be playing any video, I will let you know, and before playing it, so that it will run smoothly, I will inform you. Okay. So microorganisms, these are the agents of biosafety, basically, right? Why biosafety is required in the laboratories? Because this is again a very important things, the biosafety precautions and other measures which we have to take care. Why biosafety practices are important? The first important thing is protection, because when you are taking care of yourself, your colleagues, your students, your researchers, so protection become a very important entity, very important things. So why safety is required for the protections. Okay, then comes your, it is required to protect from protection of the students. and of the faculties. And the very important one is staff and environment. Again, this is again very important. Whatever the agents you are processing in the labs, as well as the waste generated from the labs, and any sort of the effluents and any sort of the things that is generated from the laboratory, it should be discarded and disposed properly. So why biosafety practices are required? It will be protecting us, the students and faculties, staff members, and the very important crucial component is environment. So if you are in a lab, how you are going to discard your waste, how you are going to discard your any of the laboratory effluents, any biological material, if you are utilizing in the lab, how you are going to discard them. That is basically, that will comes under the biosafety practices, right? So this again is very important biosafety practices. And what is the journal requirements? The journal requirement is knowledgeable instructor. Knowledgeable instructor should be very, very important thing. For example, if I am not able to make you understand like how you have to work in biosafety laboratory, so it's of no use basically. So it's of no use. So knowledgeable instructor plays a very crucial role in deliberating and demonstrating and guiding the researcher and the students in the laboratory, right? Then comes knowledgeable students. Whatever the guidelines and the precautions a faculty member is providing to the students, so you have to follow those. One knowledgeable students plays a very crucial role in maintaining the biosafety in the laboratory. Aware of potential hazards. Again, this is very important, the crucial, because you should be knowing about the potential hazards and any other things that is coming out from the lab. For example, if this chemicals or the reagent, it is causing the deleterious effects, the student should know. For example, this dye is causing the mutation, student should know. And because we have to maintain our laboratory in such a way so that the proper and the properly the displays, precautions, guidelines, all the SOPs, and all the documentations that should be maintained properly, right? Proficient in practices and techniques. So proficient in practices and technique means an individual should be highly proficient in working with the tools and technologies. So, for example, you can't do the mouth pipetting in the laboratory, right? So an individual should be highly proficient in practices and technologies, right? So this is again very much required in terms of handling the instrument, taking care of the any of the instrument that can pose the danger. Okay, this is again very important when you are working in any of the hospitals, medical laboratories, and other things. So category wise, it has been categorized by the competent agencies, that is category number, waste category, and how you are going to treat and how you are going to dispose those things. For example, category number 1, human anatomical waste. So it is basically the human anatomical tissues or the waste, how they can be discarded. So treatment and disposal. Incinerations means you will be heating or elevating the temperature above 360 degrees centigrades, and in the deep burial you can dispose of all these wastes, right? So human anatomical waste, for example, your human tissues or any kind of the body parts, and any surgical parts from surgeries, basically some of the carcasses, etc., how they can be very important this waste, and they should be treated properly. And then come the category 2. It is animal waste in veterinary sciences, in veterinary laboratories. So animal tissues, organs, body parts, carcasses, bleeding parts, fluid, body and experimental animals, whatever it is being used in the research work, right? So waste generated by veterinary hospitals, colleges, and discharge from the hospitals, animal houses, veterinary hospitals, etc., they should be properly incinerations and dew burial in some of the cases. Otherwise, they should be burnt out properly at high temperature, because tomorrow I will be taking you some of the practical sessions on a type of the cancer that is caused by viruses. So we have the government project also that is being run in the laboratory itself. So I will demonstrate you like what sort of the precautions, or what sort of the waste is being generated from these things, right? And then comes your category number three. So category number three includes all the microbiology and biotechnology wastes. So microbiology and biotechnology waste, for example, the waste from laboratory cultures, stocks, or microorganisms, right? So vaccine production company, whether it is live or vaccines, human and animal cell cultures, which is being utilized in research, and infectious agents from research and industrial laboratories, waste from production of biologicals, toxins, dishes, and devices used for transfer of the culture. So they can be properly autoclaved, they can be microwaved, they can be properly done with the incinerations, right? And category number four, waste sharps that is being utilized, which includes needles, syringes, scalpels, blade, because they are very crucial in the laboratory. How they should be discarded, they have the proper protocol, and these can cause cuts, punctures, and this includes both used and unused sharps. So they should be properly disinfected with either with chemical treatment, microwaving, and shreddings. Okay, so this is basically the waste sharps that can be properly discarded and properly cutted first, then go into the autoclaving and other sections. Then comes your category number fifth, which includes, expired drugs, cytotoxic drugs, and discarded medicines. How these waste, which comprises of outdated and contaminated and discarded medicines, there should be the incinerations and at the rate of properly discarded, and drugs disposal in secured landfills in this way, because drug resistance can be a very important thing which is now considered as the hidden danger for the human society and the civilizations. So this drug resistance is already, we can say, if it would have been infectious, it is the already a pandemic, sorry. So drug resistance is posing a threat to the human civilizations. Then soiled waste, for example, in hospitals, the linen, the bed sheet that is contaminated with the blood, catheters, dressing, soiled plaster cards, lines, bedding, other material which is being contaminated with blood, right? So they should be properly autoclaved and microwaving, and incinerations, they can be properly done in this way. Then category number seven, solid waste. So the waste that is being generated from disposal items other than the sharps, such as tubings, catheters, intravenous sets, etc. So this disinfection can be treated by chemical treatment, autoclaving, microwaving, and mutilations, and the shredding, etc. Shredding means cutting off liquid waste that is being generated from the lab by cleaning, washing, housekeeping, and disinfecting activities. So disinfection by chemical treatment and discharge into the drains properly, it should be done. Then incineration ashes, the ash that has been left from incinerations, that should be properly discarded. And the chemical waste, chemicals that is being generated and produced from biologicals, chemical used in production of biologicals, chemical used in disinfections, right? So they should be properly chemically treated and discharged into the drains for liquid and secured landfills, right? So this was basically the category-wise, the category-wise biomedical waste, or the waste material that is being generated, and how they should be discarded, and what are the various waste that can get generated, right? Now, for example, you are a lab professional, and in the laboratory, definitely you will be requiring some sort of the machine, some sort of the instrument, basic infrastructure of the laboratory and other things. So what are basically the basic infrastructure and other things that is there, right? So we will be discussing about all these things in this presentation. So the general requirement, that is biosafety levels. So biosafety levels are nothing but these are basically the levels of containments. We can say biosafety levels of containments, we can say, and different biosafety cabinets and instruments that is there. So general requirement biosafety levels, BSL-1, BSL-2, BSL-3, and BSL-4. Four levels of biosafety are there. And laboratory practices. Laboratory practices are nothing but when we are walking, and these can be, the safety equipments which are called as primary barriers, and this secondary barrier is basically the how your laboratory should look like, right? So biosafety level, first one, these are the requirements. Then laboratory practices, how you are working and how you are behaving and how you are following, how you are documenting into the lab. That is basically the laboratory practices. And safety equipments, your goggles, your eye shields, your mask, your gloves, these are basically the primer barriers. So primer barriers are nothing but they are basically the safety equipments, and the secondary barriers is the design, facility design, and the construction, how your laboratory has been prepared, in which directions your rooms are there, in which area you are working dedicated. For example, where is your autoclave area, where is your microbiology lab, where is your autoclaving lab, where is your dark room, where is your PCR room, where is your RDT room, where is your sample receiving rooms, right? So this is basically a requirement of all these things. So when we are talking about biosafety cabinets, so biosafety cabinets, and this is required. So biosafety cabinet type 2B2 and type 3B3. So these are the different types of the biosafety cabinets that comes for the working, right? And when we talk about the personal protections, that is the secondary requirements, which includes your PPE, personal protective equipments, your gloves, lab coats, eye and face protections, and the pipetting devices. So pipetting devices is also required. So these are the levels of biosafety. You can see there are basically the four levels of biosafety: BSL-1, BSL-2, BSL-3, and BSL-4. So with the increasing order of this thing, so BSL-1, BSL-2, BSL-3, and BSL-4, they have been designated and designed in such a way, depending upon the work you are doing. For example, you have just the basic work, then it is non-pathogenic, and we are not at all utilizing the other things, right? So it is basically the BSL-1, and the BSL-2, agents associated with human disease but unlikely serious hazards. So this is BSL-2. BSL-3, indigenous exotic agents associated with human disease and with the potential for aerosol transmission. And BSL-4. So BSL-3 and BSL-4 are the one of the highest level of containments, right? In India, there are so many laboratories that are working on in BSL-2 and BSL-3 and BSL-4, right? So BSL-3, basically, when we work on those disease which is highly infectious, they can transfer from one individual to another, as well as the disease which can be threat for the workers who is associated with other people. BSL-4. And these are dangerous. For example, any pathogen or any of the viruses or any of the disease, it is emerging for the first time, right? So how they emerges, how they works, right? And dangerous exotic agents that can threaten to the life itself, right? So this is basically the levels of the biosafety which is there, right? And basic steps to consider. For example, now if you are working in molecular biology laboratory, because in molecular biology, there is the demand of this thing, Just a second, dear students. Okay, so now this is the demand of the future as well as the current scenario. How you are designing a laboratory which require PCR related work and molecular biology related work, RDT, when you are doing, right? So this is the segmented area. This is the physically separated laboratory that should be there in the laboratory, right? That should be there in your infrastructures. So what are the basic things that we have to consider when you are establishing any of the laboratory with respect to this molecular biology? So the first one is specimen collection as per the instruction for molecular diagnostics, some of the research oriented. It should be a separated area, very, the separate area, dedicated area where the sample collection and sample storage as well as sample labeling, etc., is being done. For example, you are working on virology, you are working on microbes, you are working on pathology, you are taking the blood samples, and also the precaution should be taken while doing all these things, right? So specimen collections and receiving area, it is the way how you work. And then molecular grade reagents preparations. For example, you have to do the primer reconstitution, you have to do PCR, you have to do RT-PCR, you have to aliquate stack DNA polymerase. It is a different area. So I will be showing you the laboratory, don't worry, because there will be the laboratory video demonstration, and we have already all these laboratory dedicatedly working with respect to the work allotted. There is no work sharing profile of any of the laboratory. If it is for reagents preparation, that area is dedicated only and only for the reagents preparation. No other works should be done, should not be done in that particular area. So this is molecular biology grade reagents preparation area. Then comes your DNA, RNA, enzyme, DNA, RNA sample inactivations, pre-processing steps. So nucleic acid isolations, extraction, pre-inactivation, and pre-processing steps can be done over here. Then preparations of the multiple copies by cDNA, or preparation of the multiple copies of the targets, that is called NATs, nucleic acid amplification technologies, right? So, my dear students, all these things are going smoothly. Just for your confirmation, put in the chat box so that I will be assuring that you are listening properly and all the things are going smoothly. Just put in chat box yes or no, or any thing, because at the end of the presentation, because this will be open for the discussion, and you can ask any of the queries or the questions which you are having, right? Okay, fine. Okay, I'm getting the good response. So we will be preparing the multiple copies of the targets, that is called NATs. This NAT technology, that is nucleic acid amplification technology, is basically the latest one, and where we are targeting the DNA and RNA, right? So this is how we work, and this is how your area should be dedicatedly allotted. And then results interpretations that can be done by electrophoresis, phage, reverse hybridization, southern blottings, and technologies. So A, B, C, D, E, these are basically the different laboratories dedicated to their work, and it should be physically separated. There should be no air contamination from one area to another area, and there should be airlock system, or it can be separately done, right? And this is called as unidirectional flow of the work. So whatever the work you are doing, that should be done unidirectionally, in the single way, in a single direction or a single way you have to work, right? So this is how we have to work, right? And see, this is a very beautiful presentation of the BSL2 lab with BSL3 practices. For example, you're working in clinical research, in health sectors, in medical startups or in R&D, you will be receiving and labeling the samples. So that area should be properly labeled separately. Then they should be physically separated with airlock system, right? These are called airlock. Airlock means no air should get, exchanged between this one, right? And then reagents preparation laboratory, area one. This is called area one. That is where you are preparing the reagents. It should be equipped properly with horizontal laminar airflow with positive pressures. Then sample labeling in 100 class clean room, then airlock, then amplification. So this is how your laboratory should look like when you are planning to any other startups, or you are working in somewhere, right? So this is how we work, right? And there should be dedicated equipments and pipettes, as well as there should not be any transmission of or other or agents of any air or some contamination. It should not be transported from one area to another one, right? So when we talk about handling of the blood sample, handling of the clinical material like urine, tissue, biopsies, some of the environmental samples, etc. So there should be, dedicatedly area. For example, sample, it is coming. You should not go into the reagent preparation area with the samples. Okay. When you are working on molecular biology, cell biology, recombinant DNA technology and all, so it should be having the dedicated area. Then you can see, because we will be demonstrating you practically on 6th and 7th of August about the magnetic bead based methods, silica column methods for hepatitis C, cancer markers, as well as the cancer that is caused by some of the viruses, how they can be characterized by RT-PCR technologies. So we will be talking into more details about all these contents, as well as other things there itself. So reagents preparation, the mass mix preparation, this is basically the area that is dedicated for in molecular biology, this RDT, as well as PCR based or RT-PCR based work required this reagents preparation and mass mix preparations. Then amplifications or and detection. So amplification can be done in the PCR, and the detection either in agrogel electrophoresis or in other components. Fine. So this is how we used to work, right? So I will be talking more of these things later on in this presentations, right? And today, because I want to make you familiar about GLPs, so that from tomorrow onwards, we will be talking more about the cancers markers, genes, as well as the practical things that we are about to do, that we are going to do over here. So prevention of occupationally acquired infections. Again, it is again very important. It means that wherever you are working, whatever the occupation you are doing, if it is related to your, the handling of the samples and detecting of any of the things. So we acquired infections from here, right? For example, you are working in the hospital. So there can be so many nosocomial infections you can get, right? If you are working in the pharma industries, so drug resistance can be the factor. If you are working in some of the chemical factories, chemical industries, so fumes and otherwise the poisonous gases can be there. If you are working in the laboratory, the incidents or the accidents that can happen due to the sample leakage, pricking of the needles, inserting of the samples from one vial to another vial, and may enter into your skin through cutting and inhaling. So these are basically the occupational hazards, and how we can prevent all these occupational hazards that we are going to discuss. These are rare, not rare, but it is medically very important, and it is significantly very important, because at the time we might not be knowing, but these symptoms, signs and symptoms we will be getting later on, right? So when we are talking about these occupationally acquired infectious agents, which we acquire from occupation basically, so approximately 5.6 million workers in healthcare and other facilities, they are at a risk of bloodborne pathogens like HCV, HBV, HIV, cytomegala, some sort of other infectious agents like herpes, etc. They can be transmitted at the point of, walking, as well as when we used to work over there, right? So responsibility for safety, why basically it is required when we talk about an individual's safety in the laboratory. So we have to strictly follow policy. Okay, we have to follow all the policies. Where my voice is breaking, my dear students, or some student. I got a message from a student. Am I audible clearly, or some problem is coming? Hello, because I think from my side, internet is very much clear. So yeah, so many students, I got message that it is clearly audible. So Shrishti, you can check your internet connection. You can reconnect. I think there is problem at your end only, right? I think for rest of the students, I am clearly audible. And it's clearly audible, right? So you can check the internet at your end and let me know. Fine. So demonstrate proficiency. Proficiency technologies, as well as the proficiency in terms of research and diagnostic, means how much accurate an individual is, how much accurate a person is, or the scientist is, or the technician is. So that is basically the proficient. Receiving appropriate training, it is again a very important part of any of the occupation. If he or she do not know the work properly, it's of no use, right? So we have to receive appropriate training programs and report incidents. For example, if any kind of the accident is happening in the lab, we have to report that incidents, right? And then participate in medical surveillance, because when we are working in any of the laboratory, if any of the individual is protected for HBV, hepatitis B virus, so protective HBV titer should be there in an individual, so that it should be protected from this liver infections or hepatitis, any kind of the hepatitis infections. So we have to participate as an individual, as a setup, as a team in medical surveillance too, right? So this is again very much important when we talk about all these stuffs. So what routes of transmissions are likely to occur in laboratory worker? Again, it is a very important question. Basically, the routes of transmissions, right? So routes of the transmissions, from which routes, or what are the various causes of the transmission of these infectious agents in the laboratory workers. So routes of the transmissions, how it is going to affect and how it is going to happen, right? So route of transmission, these are basically the route of transmissions. Inhalation, some students, some researcher, even some of the faculty members, they used to still practices the mouth pipetting, which is not at all recommended. So inhalation means when you are inhaling any of the chemicals, reagents, compounds, fume, some of the cultures, vapors, etc. So this can be the route of transmission of these infections in the laboratory, right? Cuts from contaminated items. So an individual can get the cuts from contaminated item. It can be like the slides. It can be the needles. You can cut from the iron of any of the machine, any of the instruments. So these are the routes of the contamination also. And contaminated hands to mucous membranes, right? because the entry of any of the etiological agents or any of the pathogen or any of this thing is going to happen when there is basically from hands to mucous membrane and splash to mucous membranes. If any of the leakage is there, then splashes to the mucous membrane can happen. So contamination of non-intact skin. So non-intact skin can get contaminated because of these practices. So practices as well as how you are performing in the laboratory, it makes the things very, very important for all of us. Contamination of non-intact skin and ingestion, right? So this can also be done. Then ingestion. Ingestion means when we are this RTI, that is respiratory tract infectious agent, some of the infection that is because of the ingestion or inhalations that can upset your abdomen, and because of the gastrointestinal infections, there are many, right? Then root, we have already discussed. Then the potentially infectious agents. What are the various agents that can be potentially, very hazardous? Some of the fluids, some of the tissues, some of the biopsies, some of the other material, how they can be, plays a very crucial role in fact in transmitting the disease as well as, infecting the other materials. Semen, when you are working in any of the biological laboratories in hospitals, in R&D labs, in diagnostic labs. So these can be some of the, the materials or the samples types that can come for investigations, like semen. Semen in the laboratory can come for semen analysis from the cases which is for infertility, and these are the things that can infect an individuals if they by chance will be touching them, or if there is any cut or wound in an individuals, they can go there. These are basically the source of the contamination, and any kind of the material that is basically coming from the clinics or from the human origin or from the animals, they should be considered as highly infectious. They should be considered as highly infectious, right? So semen is the potential to infect an individuals through cuts, wounds, and other means too, because they can carry some of the pathogens too. Vaginal secretions. So potentially infectious material. These are basically the body fluid itself. They can be source of materials. And we will be demonstrating you practically also from tomorrow onwards how this can be done and how they can be processed inside the laboratory. Then comes your pleural fluid. You know pleural fluid. Any participant, can you please tell me what is pleural fluid? You can put in the chat box. What is pleural fluid basically? Pleural fluid, how, from where you used to get this pleural fluid? Okay, fluid from lungs, absolutely right. Fluid from lungs and fluids in the lungs. Okay. So pleural means in the cavity, basically, when we talk about the lungs. Okay, so lungs are responsible for the exchange of the air through by means we can, inhale, etc. Right? So this is sometime the cavity of these lungs, they are called as the sac also. So this fluid is basically present over there. So pleural fluid is the fluid of the lungs, sac, as well as outer lining of the lungs itself, right? And the pericardial fluid from the heart. So pericardial fluid is basically the fluid that is present in the heart and the outside the layer of the heart itself. So these are called as the pericardial fluid. Peritoneal fluid. Peritoneal fluid means the fluid of the knee, joints, etc. These are peritoneal fluid. Amniotic fluid from womb of the mother, fetus when it is developing. amniotic fluid is that fluid where so for so many investigation, this amniotic fluid used to come. Okay, so amniotic fluid can be utilized to screen out any of the defects of the newborn baby, which is about to develop, or which is developing, as well as to study the various syndromes and other defects in an individuals that can happen, right? So that is basically the amniotic fluid. And saliva, when we talk about dental practices, dentistry, these BDS, that is, or the doctors, dental doctors, they are taking care of our oral cavity, and in the oral, basically, so many problems are there. When you will, culture any of the fluid or any of the dentures from the contaminated tooth or this one, you will get the crores of colonies of the bacteria. So saliva is a very potential and highly infectious source of the material, or we can say the infectious fluid that can cause a that is containing uh, crores and crores of the colonies inside our mouth if the proper dental hygiene is not practices, right? And any body fluid with visible blood, any of the material that is with the visible blood, whole blood, some of the fluid, some of the tissue, and some these can be infectious materials as well. And okay, very importantly, when we talk about how does exposure occurs, so how these accidents happens inside the laboratories, how it is going to happen inside the labs. So this exposure can happens because of the needle stick injuries, cut from other contaminated sharps, scalpels, broken glasses, etc. inside the laboratories, right? And contact of mucous membranes, that is eye, nose, mouth, or broken cut or abraded skin with contaminated blood, right? So the infections can happen, or they can be transmitted from these sources also, right? From eye is also the source of contaminations. This fluid that is present in the eye vitreous, as well as this can be source of infection. Nose and mouth, they contain so many the shedded of viruses and even the bacteria also, and or broken cut or abraded skin with contaminated blood. So this is how this has happens, right? Biosafety, what actually the principle of the biosafety, it provides the level of containment. So what is basically containment? Students, can you tell me what is containments or level of containments? Why we call it as a containment? So again and again, I am just talking about the containment. So what is the containment? How it works, and containment means what? What actually you know from this word containment? Okay, some replies have come. So isolation, perfect, right? So isolation. So when we talk about the biosafety, these increasing level of containments, that means that when we are talking about the containments are nothing but these are basically the separated area from where we can prevent the infections. Containment zone during no sterilization. It is not a sterilization basically. Containment means, for example, if there is spread of the disease in a particular location or a area, right? How you are going to prevent it? So you are going to contain that area. You are going to restrict that area for the entry of any of an individuals. That is basically the containments. During COVID, you have heard so many times containment zone prepared, right? So containment zones are nothing but these are basically those zones which are totally isolated, right? There is no entry of any of the individuals over there because they are basically the contaminated one. So yeah, so that's why Facility of isolation to protect either the sample or the person or the both. Very good. Rohit, Rajan. Okay, so containment. When we talk about the laboratory, basically, containments are nothing but containment means when we are restricting any of the contamination to a particular location or a particular area. And the biosafety is the best example of making this containments. So this BSL1, BSL2, BSL3, BSL4, these are nothing but these are also the level of containment, right? Personal and environmental protection. So how personally and when we talk about the environment, how it is going to, protect, how we are going to get protected, personal and environmental protections, right? Risk of infection from percutaneous exposures, right? So this is again a very crucial and a very important. You can see the type of the viruses and per ml of the serum, how much virus is present in a single ml, that is in 1000 microliter of your sample, or in just a single tiny droplet or in a nuclei, how much viruses can be there. And you can see 10 raised to power to 10 raised power to 2 to 10 raised power 8 viruses. It can be in one single ml of serum. And what is the percentage of risk of infection? The very crucial is HBV, hepatitis B virus, viral infections, which is contributing to 30% of the risk of the infections, right? Then hepatitis C virus, HCV, 10 raised to power 6 viruses can be there in per ml of the serum, contributing 3% of the infection. Then HIV, 10 raised to power 3.3% of this infection they can contribute, right? Universal precautions, what we have to take. For example, you don't have any idea about what is this biosafety all about. So universal precautions is treat all human blood as well as other certainly body fluids as if they are infectious, right? So this should be observed in all the situation where there is a potential for contact with blood or other potential infectious agents, right? So when we talk about the laboratory practices and techniques, so laboratory practices and techniques, it should be restricted during experiment, restricted area to the lab. No one is allowed, no one should be allowed basically inside the laboratory. So the area should be totally restricted during experiments, and immunocompromised individuals excluded. Immunocompromised individuals means those who have weaker immunity, those who have the lower immunity. For example, HIV patient, these are also called as immunocompromised individuals. The individuals undergoing dialysis, they are also immunocompromised individuals. Pregnant female, they are also immunocompromised individuals, right? And comorbid individuals, those who are suffering from the lifestyle disease or other disease, etc., like diabetes, high blood pressures, cholesterols, and these individuals can be immunocompromised. So their entry, if it is possible, restrict their entry, and there should be no entry for all these individuals. And if they are required in the lab, so they should be proper, they should properly follow all the guidelines as well, right? So this is why immunocompromised individuals are all above, right? Yes, organ receivers, definitely. Those who are organ receiver as well as the donor also. So donor as well as the receiver of the organs, they are also immunocompromised. And during the immunocompromised situations, there is basically the decrease of the immunity to very, very less amount, so that any of the infection can occur, right? Doors kept closed during experiments. So this is the general things that we have to do. Biohazard sign posted. See, proper washing we have to do. Do not eat. These are just the basic thing. I guess you people know very well. So do not eat or drink in the laboratory, and then proficiency of the handling of the machines. How I have to basically handle the this micropipette. How I have to utilize this thing in microbiology, right? So we have to use the mechanical pipetting devices whenever we are working on these things, right? So laboratory practices and techniques. So proper for this needles and sharps, we have to properly keep the waste bin and do not bend or resheet or reuse syringes or needles. And you have to use the sharps containers properly. See, this can be highly dangerous, and where there is basically coming out of such type of the needle prick injuries can happen, right, to the students. Laboratory practices minimize the creation of the splashes. So any kind of the release of the aerosols and any the splashes and leakage, it should be avoided in the laboratory, right? Decontamination of the workspace surfaces. So you have to properly decontaminate the workstations. So how you used to do in the lab, my dear students? Can you tell me how you, the sterilization of your biosafety or your laminar airflow is being done by you people inside the lab? Can you just put in the chat box how you used to do? Autoclave. We can't autoclave biosafety cabinet, na? We can't put the biosafety cabinet into the autoclave. Basically, there can be some of the things that can be autoclaved, but not for all. Spraying alcohol, ethanol, fumigation, alcohol by using 70% alcohol. So that's basically some of the reasonable answers. UV sterilization plus 30% alcohol. Heap HEPA filter. It is not heap filter. It is a, yeah, correct. HEPA filters. So it is equipped with the biosafety cabinet or in a laminar airflow, these HEPA filters, which is utilized over there. So yeah, 70% alcohol. For example, if you are working on the DNA, RNA based things, so what precautions or the what are the things that you are going to use? If you are working on the nucleic acid isolation, PCR setups, and recombinant DNA technology, what precautions you are going to take? So see, you have told me about all these are 90% are microbiology tools and techniques, and some of the techniques in microbiology, they can be commonly used. So can you tell me, my dear students, what are the, the basic other uh, precautions that we have to take care when we are doing the molecular biology grade work, PCR or RT-PCR, DNA isolation, RNA isolation, etc. Can you please tell? Any answers? Okay, some of the answers I got. Okay, maintain temperature so that they do not denature and proper handling. See, for example, you are isolating the DNA from blood for any of the cancer in biosafety hood. So what precautions you will take? Can you tell? Hmm. Using enzyme. Which enzymes you will be using? Using enzyme. How these enzymes can be useful for precautions, for sterilization, and which enzyme? anticoagulant, EDTA, anticoagulant. Any other answer? Here I will tell you like what are the various precautions we have to take during molecular biology grade work. First of all, the most important enemies of DNA and RNA, nucleases, enzymes. These are the most important enemies when we work on the DNA RNA isolation processes, right? So what precautions we have to take when we are working on DNA RNA? So first of all, we have to use nucleus-free water, NFW, which do not contain any nucleases. UV treatment, it should be given. Then absolute ethanol and sodium hypochlorite solution, it should also be given. And there are some of the formulation like enzyme that is called ZAP solution, or some of the uracil and glycosylase. These are the enzyme that can be utilized to get rid of these, contamination that is because of your secretions from sweats, etc. Right? So this is how we can take precautions while we are performing the molecular biology grade work. And aerosol barrier-free tips, DEPC solution. The DEPC is nothing but diethyl pyrocarbonate solution. It is a solution that is nucleus-free water. It can be utilized, right? So this is, NFW. This is nucleus-free water. Like you are utilizing this molecular biology grade water or distilled water. In the same way, nucleus-free water used to come for molecular biology grade. And this we are going to do the practicals, and you will be learning so many things practically from make internal, learn to upgrade, so that you will be familiar with all these things. What is NFW? Because on 6th and 7th, we have the practical on this one only. Okay. So this is nucleus-free water, NFW, which is being utilized in PCR, RT-PCR, in recombinant DNA technology, in RDT, and other things too. So this is how we used to work, right? So decontamination of work surfaces before work, after spills, end of the day, we can do with these things. Okay. Next is safety equipments. That is again very important. Have you heard about the word ergonomics? Ergonomics. Have you ever heard the word ergonomics? Ergonomics, basically, we call it as laws of doing work. For example, you cannot construct a 10-foot this biosafety cabinet for an individual or for a technician or for the student who is only this 4.5 feet, right? So it should fit according to the ease as well as the stature, physical appearance, and energy limitation of individuals, not about other things too. So that's why this is called as ergonomics, right? And safety equipments, protective clothing, lab coat, decontaminated gloves, eye protections, and safety equipments. For example, you're using the centrifuge safety cups, right? And leak-proof transport containers. Primary barriers, use biosafety cabinet class 2 for walk-in infectious agents, aerosols and splashes, and a large volume, high concentration, we can utilize this class 2 cabinets. Then these are just basic. I think it is present in all the colleges and university, LAF, clean benches, the product protection only, hazardous aerosol, it is going for exposure of the operators, and this laminar airflow hood and biosafety cabinet. These are basically the two important components of any of the laboratories if they are working on highly infectious materials and samples. And the very important thing is how this air circulate, how this air works in this, and how we can have the airflow patterns. So air enters through work opening, and 30% will be the exhaust, and 70% recirculates. So laminar flow, this is how we work. And when we talk about the biological biosafety cabinets to contain the aerosols, so you can use biosafety type 2, B2 or type 3, B3 biological safety cabinet, because there will be no recirculation of the air in biosafety cabinets, right? So this is how we used to do. And effective use of biosafety cabinets, pre-planning, use only material that is needed for procedures. You cannot put so many things at a single place, right? Arrangements of the clean and contaminant means these are just the basic of biosafety. You should not move in and out so many times your cultures and other types of the media. Properly minimize room air current from walking near cabinet, opening doors, and room air vents, etc. Right? How do you dispose of pipette while working in biosafety? Pipette disposal. Do not use container outside. That we always do, and do not use upright container inside the biosafety. That is basically the recommendations because of the air currents and the air flow over there, right? And use horizontal tray with disinfectant or dispose of in a small biohazard base for autoclaving. Secondary containment, how your laboratory should look like, that is again a very important component. Secondary laboratory facilities, lockable doors, proper hand washing sink, and which should be designed for easy cleaning, and non-fabric furniture should be utilized. Biosafety should be properly installed, backup with the generators or UPS or inverters, and you should be having the proper IWAS stations. Any kinds of the accidents can happen in the lab, and good illusions. These are the chemical disinfectants that we can utilize, right? Glutaraldehyde, formaldehyde. I'm just taking the basics today so that we can finish it up today as well, just in the introductory sessions. And from tomorrow onwards, we will be focusing more and more on this cancer biology, various types, diagnostics, and the methods for diagnostics, treatments, new updates in all the fields, right? Okay, so we were talking about chemical disinfectants. So chemical disinfectants includes glutaraldehyde, formaldehyde, chlorine compounds, iodophores, phenolics, alcohols, quaternary ammonium compounds. Agent efficiency, that is how basically these things works when we are talking about its efficiency or the efficacy of working in any of the environment. Target microorganisms, how they are going to work on any of the target microorganisms. Concentration, at what concentration they are going to work, right? Either we have to dilute, we have to concentrate, amount of organic material that is present in that thing, type of material to be treated, which type of the material we have to treat, type and concentration of germicide in them, contact time, how much incubation you are going for these things to do, then temperature, then pH, then humidity. These also are very, very important when we talk about agent efficiency. For example, glutaraldehyde, all these things, how they works. Basically, resistance to germicides, right? So resistance to germicides, when we talk about the various chemicals and the reagents and how they are getting resistance to these germicides. And you can see like a least resistant fungus to prions, that is viral copies in the viruses, how they are getting resistance to these things. Vegetative bacteria, lipid viruses, non-lipid viruses, mycobacteria, bacterial spores, prions, etc. They are there. Chemical disinfections. So chemical disinfections, high level sporicidal, glutaraldehyde, intermediate level like Mycobacterium tuberculosis, chlorine and phenolic that can be utilized at intermediate levels, right? At high levels are glutaraldehyde, which is sporicide, even they can destroy the spore also. For example, bacterial and all, how you are going to work for them is glutaraldehyde, etc. Okay, so this is how they works, and the low level based are quaternary ammonia and others, right? Just a second, my dear students. There is okay. This is how it works, right? Chemical disinfectants and disposal of biosafety waste. How you are going that all these things, because tomorrow we will be demonstrating you and we will be taking you video to the labs, video to the laboratory. Okay, we are going to take this to the laboratory tour, disposal of biosafety waste, because in any of the laboratory, this biomedical waste management treatment and good laboratory practices they are going to do, right? And we will be talking more about the biosafety components as well as other things, uh, and how this waste can be put into the autoclave bags and how they can be discarded properly, right? And in which manners and in which way they should be discarded, right? I think these things we will be demonstrating you practically tomorrow, because you have chosen this subject, this cancer biology, for your online training program. So we are doing the cervical cancer, we are doing the blood cancer, we are doing the liver cancers, how these can be discarded properly, that we are going to demonstrate you in the laboratory itself tomorrow through video demonstration, right? So this is how this autoclave bags and other things should be properly sealed and taped and then discarded, right? So I have so this is how lab attire. You should remember the following: no open toed school, no short unless a lab coat is used, retain hairs. So this is just the things and the habits, I think all you know. And tomorrow, because I think for another 10 5 to 6 minutes I will be taking you about the cancer epidemiology. Before that thing, any sort of the queries or the questions, if you are having, this platform is open to all of you now. You can ask any sort of the queries up to here. So you can put in the chat box. Okay. So because this is the first day we are interacting, right? And because we have other colleagues and the fellows who will be taking the sessions, not session, basically some of the practical they will be demonstrating, as well as I will be taking the sessions along with them, our junior scientists and research scholars. They will be taking care of this program too, indirectly, basically, right? And now, if you have any queries or the questions, you can put in the chat box. Any doubts? Because this was just the basic class, general lab practices and other things. Basically, when I am also a biotechnologist, and I will also be sharing you some of the aspects and other things regarding startups, industry-oriented training programs, as well as how to develop your own laboratories in diagnostics, in virology, in RDT, some other things also, because we have our own organization. So it is going to add on so many things for you people, because I started the first laboratory of swine flu and H1N1 and bird flu and gene-based diagnostic in Uttarakhand. So I will be sharing that experience to you also, so that you will be getting so many ideas for the startups, right? So no questions, I guess. So can we end up for today, so that because I have a very important assignment today at 5:20 p.m. onwards, right? And tomorrow, because this program is going to run from 4 to 5:30 on a daily basis. But today I'm leaving you at 5:15, and 10 minutes more we will be taking tomorrow, right? So any sort of the queries or the questions you are having, you are most welcome. I will be happy to answer. Otherwise, we will be meeting tomorrow. Okay, so tomorrow we will be talking about the basics of cancer biology, and I will take you to the laboratory tour tomorrow in the beginning itself, and we will be talking many more things tomorrow also. Fine. So thank you very much for your patience, as well as joining with me for the starting of the cancer biology course. Okay, so so you understood so many things. Fine. Okay, so we will be meeting tomorrow then. Okay. And if you will be having any doubts or any clarifications, we will reply tomorrow also, no problem. Thank you very much. So I am signing off now, and we will meet tomorrow. Thank you.
मैंने वो वाली बात देखी, पता है कल लोग क्या बोलते हैं, मार्केट गए तो मार्केट में ये है स्टूल है यहाँ
एसी ऑन कर दो। हम आए तो काम कर दो।
बिना एसी के रहते हैं हम।
नहीं एसी आपको चलाना है।
क्या ऑन है रिकॉर्डिंग
एसी में रहा करो मैं
एंड क्या मीटिंग क्या कर रहा था
जो ये गोल अपूर्व मैं या ठेके क्यों नहीं
ये रिकॉर्ड क्यों हो रहा है? अच्छा रात हो गई,
12 रात हो दिन आज 6-7 बजे का रात है।
बारिश हो रही थी, डर लग रहा था मुझे, दिख रहा है डर लगना जरूरी है।
क्या हो
कुछ कुछ
ठीक है मतलब सीरियल नंबर 1 2 3 4 हम यहां भी सीरियल नंबर 1 2 3 4 लेकिन सीरियल नंबर पॉजिटिव का लेना ना तो हम पॉजिटिव ही रखते हैं ठीक है जैसे क्या है एक नंबर में वो खाली कैसे निकल गया गलती से पता नहीं चला कौन सा नंबर जैसे हमने कहा आपको
ये बहुत वाली इसमें हो रहा है
मेरे पास यहाँ से केवल कौन ले गया ये यहाँ अब बता यहाँ मैं टेम्पलेट वाला ही है ना आके रखा पर ऐसा तो जल्दी काम दे ये मेरा है मेरा वाला ये कैसे आया नहीं करता है ठीक है अब थोड़ा सा बात आ गया
वीडियो तो नहीं करना है, मतलब वीडियो को ही ध्यान कर रहे हो। अरे सब कुछ
कुछ नहीं है, वो सैंपल नहीं दिया, बट अभी कोविड कोविड पॉजिटिव का है ना सर। ये ऑन तो नहीं है,
ठीक है, पर इसको हम ऑन करते हैं।
ये रिकॉर्ड क्यों कर रहा है, ऑन है ना।
एक बनाते हैं सॉफ्टवेयर, ठीक है। यहाँ पे एक साथ एंटी मतलब सॉफ्टवेयर है, सिंबल के बाद तो आपका सॉफ्टवेयर है, ठीक है। इसमें क्या करेंगे, ठीक है, तो दिक्कत यहाँ यहाँ पे न्यू बना रहे लगाए हैं, वो हमें याद होना चाहिए। तो हमारे पास रहता है 36 वाले से शुरू करें, 72 वाले हमने पढ़ रहे हैं 72 लॉस करेंगे, इसको लॉस ऑन नेक्स्ट कर दें, ठीक है। अब ऑपरेटर कौन सा है, तो ये नहीं, ओके। अब रिएक्शन वाला, अब बताओ रिएक्शन वालों में 10 माइक्रोग्राम मिलता है, तो वहां पे क्या कहलाते हैं, एंजाइम मास्टर मिक्स एंजाइम, और प्राइमरी कितना हुआ 11, टेम्पलेट बोला ना हमने, टोटल कितना हुआ, यहाँ पे कितना रिएक्शन वाला। अब ये सारे सेटअप हो गए, ये सैंपल है, आप सैंपल का सीरियल नंबर देखो, 1 2 3 करना है, अगर इसमें चेक करना तो ये चेक करना है, ठीक है। रिएक्शन हो, अब आ गया टेम्परेचर, जो हमने क्या बताया, देखो क्या होता है,
नंबर सेव कर लेते हैं,
आरएनए से पहले डीएनए बनेगा, तो यहाँ पे क्या किस में जाएंगे, फाइल करेंगे यहाँ पे, ठीक है। जैसे प्रोफाइल करेंगे ऑप्शन आएगा आपको प्रोफाइलिंग का, ठीक है। तो इंसर्ट आंसर पहले हमने बोला था होल्ड है ना, यहाँ पे होल्ड, ठीक है। यहाँ पे ये देखेंगे कि ये कैसे होल्ड है, ये स्टैंडर्ड मेथड है, रैपिड मेथड, ठीक है। पहला 65 से है 9 मिनट, तो होल्ड 1 होगा ये, तो यहाँ पे क्या होगा, आरएनए से पहले क्या करें, होल्ड 1 पे
बहुत कंफ्यूजिंग रहता है, पता नहीं किसको 8 9 7 9
कितना मिनट है,
3 8 सर इसमें 9 6 7
ठीक है
9
कोई दिक्कत। फिर एक होल्ड क्या बोलते हैं, तो भी क्या करेगा, होल्ड 2 में क्या बोला हमने, डीएनए प्रिपेयर रीडिंग करेगा सही से हुआ या नहीं, होल्ड 2, ठीक है। कितना रीडिंग करेगा,
95 2 मिनट्स सेव करके मैसेज करो, ओके।
हम कहीं अलग जा रहे हैं, प्रोटोकॉल के हिसाब से जा रहे हैं, हमने ये रैपिड बना रहे हैं, स्टैंडर्ड नहीं बना रहे, रैपिड बना रहे हैं, ठीक है। फिर आप देखो नीचे क्या था, साइक्लिंग था, ये साइक्लिंग कंडीशन है, न्यू साइकिल, तो साइकिल कंडीशन आ रहा है, यहाँ पे दिखेगा साइक्लिंग कितना है, 40 साइकिल है ना, देखो 40 साइकिल है साइक्लिंग, तो यहाँ पे नंबर ऑफ साइकिल कितना हो जाएगा, यहाँ पे देखो कितना नंबर ऑफ साइकिल, यहाँ पे 45 को हम चेंज कर देंगे, नंबर ऑफ साइकिल भी हम चेंज कर देंगे। अब देखो 95 पे कितना मिनट है, 95 है, ये भी
डबल 6 तूने डबल 9 अरे
फिर वो सेकंड में जाएंगे, तो सेकंड की साइकिलिंग कितना है, 60 पे कितना 30 सेकंड, 95 पे ये, और ये साइकिलिंग को नहीं लोकेशन, क्यों नहीं लोकेशन, तो केवल दिया जाए जब हमारा डीएनए इशू बन जा रहा है,
हम रैपिड मोड में तो उसमें कोई वो नहीं है,
सिस्टम
क्रॉस कर देना,
को हटा देंगे, अब हमारा आता है इक्वालिटी, तो आपका जहाँ पे रिजल्ट आ रहा हो स्लो करना है या देखना है किस पे रिजल्ट आप देख रहे हैं, उसको बोलते हैं क्वेश्चन फेज। जहाँ पे आप एक्वायरिंग करोगे यहाँ पे आप चैनल सेलेक्ट करोगे कौन कौन से चैनल में हमारा जो जिन टारगेट है वो देखो यहाँ पे टारगेट जिन कौन है पैम, इसमें पैम ग्रीन है, यहाँ पे एक और चैनल ग्रीन है और देखो टारगेट चैनल कौन सा है एनजीन, एनजीन में है एनजीन एक्साइड है ना, एक्साइड जो एनजीन को टारगेट कर रहा है वो कितनी चाहिए यहाँ पे आरडी आरपीजी, इसमें देखो आरडी आरपीजी एक्साइड, एक्साइड इसमें ऑरेंज ऑरेंज में लगे यहाँ पे। अब देखो और कौन सा है CY5 रेड हैक्स, हैक्स किसमें है देखो ठीक है, तो दिक्कत यहाँ पे आ गया सारे। यहाँ पे एक और चीज टारगेट कर सकते हैं कौन CY5 ठीक है, जो आपका रेड में तो हमने कोई जरूरत नहीं तीन टारगेट करनी
वो तो एक
वो आगे
कितने देर में फ्री
अपने पे आपका
हो?
ठीक है, ये बन गया इक्वेशन पे यहाँ पे आ गया ये लैट आउट एक छोटा सा जहाँ पे जीन टारगेट है जीन को टारगेट डिटेक्ट करेगा, जितनी प्राइमर आए हैं वो डिटेक्ट नहीं करेगा,
मतलब मैं जा रही हूँ
तो कोविड का प्राइमर लाइन यार तब शो करेगा नहीं करेगा
जॉब में मैं
नहीं करेगा सिंगल से बात है ठीक है, जो डायग्नोस्टिक कर रहा है ठीक है। अब क्या करेंगे ओके कर देंगे। अब यहाँ पे जाएंगे गेन ऑप्टिमाइजेशन, यहाँ पे आपको देखना है कोरिलेशन कितने पे है, यहाँ पे स्टार पर है ना ठीक है, हम क्या करेंगे ठीक है, हम इसको रहने देंगे और इसको क्या करेंगे ऑप्टिमाइजेशन इक्वालिटी ठीक है, इसको 3 बार पे करना 3 बार पे एडीएफ के बाद हम क्या करेंगे इसको लॉक कर दिया इसके पे 3 चैनल लॉक अंदर ठीक है, से नेक्स्ट करेंगे अब यहाँ पे सेव टेम्पलेट या स्टार्ट रन कर देते हैं ठीक है। पहले देता 2 घंटे अब देखते ये कितना मिनट है, अगर ये बड़ा काम कर रहा है तो भाई तो ट्रेन ही जल्दी देता है।
फिर ऑफर के बाद क्या ड्रॉप हो गया, लेकिन के बाद क्या करेंगे?
रिजल्ट ही देखते हैं,
हाँ हाँ नेक्स्ट
रिजल्ट देखेंगे कि क्या रिजल्ट आया
कलर के बेसिस पे, मतलब रिजल्ट कहाँ
चैनल के बेसिस पे, तो ये चैनल है इसके बेसिस पे ना
पॉजिटिव नेगेटिव
हाँ ग्रीन टारगेट येलो ग्रीन और येलो आपका आईसी है, टारगेट जीन है आपका स्पैम और टारगेट है एनजी,
मतलब वो इस पे शो कर देगा
ठीक है। अब देखो पहला सैंपल क्या था इसी था ना पीडीसी पीडीसी और नेगेटिव टेम्पलेट कंट्रोल ठीक है, दूसरे में सैंपल हम डाले थे उसको हम डिफाइन कर देते हैं, लास्ट में क्या था एनडीसी नहीं है यहाँ पे नंबर आ गया ये स्टार्ट हो गया 87 मिनट टाइम कम हो गया उसका रैपिड वाला मैथड किसी का रैपिड हो गया साइकिल से के टाइमिंग में कितने रिजल्ट हाँ 15 मिनट बाद और क्या है उसको खा हो गया प्रोग्रामिंग में यही है और सिखाने की क्या स्वामी मिलेगा कि नहीं मिलेगा पहले लिखो तो उसका क्या क्या था क्या हाँ लिख दिया हो गया
अगर हमारा काम नहीं है तो हम नहीं आते मेरे ऑनलाइन का है मेरे ऑनलाइन का है वही मैं बोल रहा वाला काम कर रहा है ऑनलाइन का हो गया मैं बोला कि सबके लिए जरूर दे देते हैं दे देंगे वो तो दे दो काम को तो दे तो उसके अंदर हो नहीं गई तेरे को किसी जरूरत मेन तो हमारा बायोस्ट्रक्चरल एक्टिविटी है ना, इसका मेन है ना क्या कंपाउंड है, हम तो वो स्टडी नहीं कर रहे हैं कि हमें एकदम कॉन्सेप्ट। और हमें तो जो एक्सपेंस बोलते हैं तो जिससे हमें सब जगह पे नाम दिया जाता है, इट इज नॉट नेसेसरी वो जरूरी है, दैट इज जरूरी व्हेन यू आर एकदम प्रिसाइज वही वाला चाहिए मुझे, वो वाला नहीं चाहिए। तो उसमें कैरेक्टराइजेशन करने का तो मुझे
मैंने कहा सर आप क्या नहीं बोल के कर जाओ, मुझे कोई अच्छे हैं और बोले ना लेसन
सर नींबू इस नींबू वो रूम में रहेगा तो वो पक जाए।
हाँ पेड़ से टूटेगा तो जल्दी पकेगा। तो सर फिर अभी तुरंत करेगा, रुकना पड़ेगा, रिजल्ट देखने पिक्चर ले लेंगे रिजल्ट का।
और तो रिपोर्ट में लगा दे
इनके मास्टर में मतलब मैं आरएनए वाला जो है ना मैग्नेटिक बीट से करा था, और पीसीआर मास्टर में एक नहीं शो करूंगी यही वाला कनेक्टर, ठीक है। मुझे बहुत करना है काम, माइक्रोबायोलॉजी के पता है प्रॉपर जो है ना रिपोर्ट वगैरह पूरा टाइम करना है नहीं, मतलब है हो गया मेरा मुद्दा इसके लिए टाइप नहीं करना पड़ा, बड़ा भी इंप्रूवमेंट जो है ना उसकी भी लगाने सकते तो है कॉपी कर लेंगे। तो दोनों आ रहे हो क्या क्या जा रहे हो क्या खाते कोई मैगी और ये क्या कहते हैं चाय में मोमोज और वो क्या कहते हैं नूडल्स, नूडल्स अच्छा था बट मैगी खाई ले तो नूडल्स भी मैगी एक ही कर नूडल ले लेते हैं और अगर 3 लोग हैं ना पेट से भरेगा फिर रात लग जाएगी या सबमिट करेंगे हाँ वो भी सबमिट कर दी जाना है मेरे को
काम अगर हुआ ही नहीं हुआ मेरा से तो क्या लिखेंगे और क्या सबमिट करेंगे, बिगेस्ट क्वेश्चन दैट
पेपर वाले में करना है पेपर डाल के ना तो रिसर्च हो जाएगा। मैं तो कहती हूँ मस्त ना तो मैं घर पे रोज सो गई दिन में मस्त आना काम से 7 तारीख के बाद तो मुझे तो सर्टिफिकेट दे देंगे तो मैं क्या करूँगी फिर मैं यार लेकिन कोई है भी नहीं घूमने के या तो मैं दिल्ली में दोस्त है मेरी दिल्ली में पढ़ाई क्लासेस चालू मुझे आ गया कंफर्मेशन पिछले से होते हैं हाँ तो कुछ तो मांगा होगा लेकिन हाँ नहीं वहाँ जाना सबमिट नहीं करना डेट नहीं दिया मैंने कुछ चेक भी नहीं तो मुझे ताने ने मैसेज करा मैंने कहा बहन मैं घर जाकर उनके लिए प्रॉपर फॉलो करूंगी और ये टाइमिंग नहीं कॉल है हाँ मम्मी ने मुझे कॉल करा ना उठाया नहीं मतलब उठाया नहीं किया मतलब बजे रात कल समझा रहे थे तो मैंने बोल दिया कि लैब में can copy of your provisional admit card select the data for computer class will be informed by mail saying classes that is being completed मैं सोच रही हूँ कि एक हफ्ते पहले जब की क्लासेस सेटल मेटल होने में ये प्रॉब्लम वाले दे दूँगी तो ड्रेस कोड क्या बोले कि फर्स्ट डे आपको जब आना मेंशन है कि सलवार सूट और दुपट्टा लेके मतलब प्रॉपर अच्छी बात नहीं है बट अब मतलब सलवार मतलब क्या वैसा थोड़ी बनवाने नीचे लगी तो कुर्ती पहन लिया मैंने चुन्नी ले ली सलवार भी बोला है पागल है क्या? खादी का कपड़ा थोड़ी पहन के स्कूल में खादी है मतलब पर सलवार कोई ना बोल रही पूरा सलवार चेंज करो कोई ना बोलेगा कितने का सलवार वो खादी सलवार की पैंट से ले सकते हैं वो थोड़ी है। अभी हॉस्टल वगैरह तो उसकी नहीं हुई है वहीं जाके जो है ये ना डेट बढ़ गई पता नहीं डेट मेरे को तान्या ने भेजा था कि प्रदूषण का डेट बढ़ गया मुझे तो मेल नहीं आया कुछ दिन कुछ टेक्निकल इशू की वजह से मैंने कहा इसीलिए वो लोग ऐसा कर दिया 5 तारीख मतलब नहीं वो तो पहले का ही था मैंने 5 तारीख देखा। अरे मॉक टेस्ट आया नीचे में मॉक टेस्ट नहीं दिया लोग ने दिया नहीं है इसमें भी इस वजह से बोल रहे थे कि डेट मॉक टेस्ट का डेट बढ़ गया होगा तो मैं बोल के देखती हूँ देना हो तो मैं देखती किसी ने नहीं दिया होगा मॉक टेस्ट हाँ मॉक टेस्ट क्या है नहीं बहुत मॉक टेस्ट उसके लिए देते हैं ताकि एग्जाम वाले दिन दिक्कत ना हो रहा है बैठ जाए ना प्रोसेस पहले सबका खाली प्रोसेस का डिफरेंट होता है सबका वो अलग और कभी कभी नहीं होगा बहुत सारे नहीं होता ना कि वो बताते हैं तुम नर्वस होते ना कैसे कितना मिनट होगा 1 घंटा होगा 80 मम्मी से बात कर ही लेती हूँ बोल देते हैं ये आने के लिए चाहिए अगर आप क्या सकते हो पर मैं तो करता हूँ उसी में एक्स्ट्रा डाल के बना लेकिन मैं मुझे समझ आया कि बीटेक्स तो हम पहले मर्चेंट की तरह यूज करेंगे ना उसको ऑयल कंप्लीट बन जा रहा है ना बीटेक्स को इस तरीके से जेल में बनाने यूज करेंगे हाँ सिर्फ बीटेक्स में हम डाल के एक्सटेंड डालेंगे और हमारे उसमें क्या क्या है वो मैम को बोलना पड़ेगा मैम डीवैक्स के साथ और ये एडिशनल चाहिए बस जैसे हम बाजार में देते हैं ये सब्जी चाहिए वैसे मैम को दे देते हैं तो ये है अगर कल आप कब तक आ जाएगा तो कल आ गया तो एक बार हम आने के पहले पूछ लेंगे अगर मैम हुआ आया है नहीं आया हम बोल देंगे कोई मैम वो चीज़ का प्रोटोकॉल वगैरह तो कुछ कर लेंगे हम तो बोलते हैं ना मैम थीसिस वाला काम करना मैम देखने जाते हैं हम काम कर रहे कि नहीं अभी मैम को बोल देंगे कि मैम उसको बीवैक्स के सिवा भी ये चीजें चाहिए मेन चीज है बीएसआई से वो तो आ जाए वो आ जाने से बहुत कुछ हम डिस्कशन इसमें सब करते हैं हमारे ये हो गया ना कि ग्लोबल टेक्स्ट होने वाला चीज मेन तो ये है ना इतना पता है
हम ऊपर कवर लेटर ना मैंने बना